This week, we're going to focus on viruses. Viruses are not going to be part of and described as living organisms. The reason for this is that it is missing many of the life characteristics that we learned about last week. Viruses are technically not cells, even though they do have some DNA or RNA, so they have some nucleic acids, and they have an outside covering called a capsid. This is a term you do need to know. The protein coat or covering is called a capsid. Now, viruses are microscopic. They are extremely small, ranging between 20 to 400 nanometers. This is very, very small. Actually, one of the largest viruses is still smaller than and somewhat close to one of the smaller bacteria. So viruses are extremely small. Now, this is one of the characteristics that viruses cannot do, and it's very important. It cannot reproduce on its own. It needs a host. It needs something else to replicate its DNA because it cannot do that on its own. It will force an infected host to replicate its DNA. So technically, it's going to take control of the transcription and translation processes. Remember, you learned about transcription and translation. Transcription is going DNA to RNA, and then translation is RNA to proteins. So in order to make these parts, now the host cell will not be doing its normal processes to sustain its life, but it will start acting like a zombie, something that's not in its own mind, and it's going to start making the viruses. And ultimately, it'll make these viruses that will kill the host cell. Okay, there are two basic theories about where viruses came from. One of the theories is the escape gene hypothesis. This is coming from an evolutionary standpoint that a gene or piece of DNA came out of a cell and then basically on its own, it evolved into a virus. What we do know from the cell theory is that cells come from pre-existing cells. Basically, if a organelle comes out, what we see in an experiment or in science, that organelle can never form into another thing. So there are problems with this idea. Again, it is a theory because it cannot be proved. Okay, ultimately, it is a theory that cannot be proved. The next one is created by God, and this is coming from a Christian perspective that perhaps it was for good in the Garden of Eden, and then it, like a lion and tiger, maybe had a different function in the Garden of Eden, and then after sin, it went ahead and the lion and tiger ate meat. So maybe in that same way, the viruses have a different function after sin. Before we move into viruses, you have maybe heard the word vaccine. A vaccine is a biological preparation. It's like a shot or a spray that is injected maybe inside you that improves the immunity of your body to fight against a certain disease or against a certain virus. So a vaccine actually came from the usage of cowpox. So cow meaning vacca in Latin. Now, a vaccine contains a weakened or non-functional virus or microbe that is injected into your body. This actually allows your white blood cells to attack it, coat it, and then basically know how to kill it. Okay? A vaccine is not an injection that will help you be cured from the virus, or the vaccine is not something that will kill the virus. No, a vaccine actually contains the virus. So if I want to get a flu vaccine, they're going to put the flu in the vaccine. Now that flu is a virus that is a weakened or non-functional virus. Now I'm not saying dead because technically viruses are not alive. So it's a weakened or non-functional virus. They inject that flu into my body and it allows my body to fight the flu, the virus. It actually gives you the virus so your body, your immune system, will fight and destroy it. Now, Edward Jenner in 1796, he's the one that you need to know. He's the one who actually used the vaccine in human beings resulting in basically a protection from smallpox. And then maybe you've heard of Louis Pasteur. He did a lot of research on microbes, bacteria, and viruses, and so on. Okay, viruses do not have 3-4 basic shapes. They have hundreds of different shapes, and there's hundreds, thousands of different forms of viruses. So some of the types you can see are spherical, circular. Some are going to be cylinder shaped. Some are going to be polyhedral with many sides. And some, if you look at the right-hand side, the T4 bacteriophage is in this spider alien shape. The first one we're going to focus on is the cylinder-shaped virus, and that is the tobacco mosaic virus. This is a plant virus that specifically attacks the tobacco plant. Now, there are other plant viruses such as the tomato mosaic virus and some others. You can specifically see with this virus, it has a protective coat, a protein coat called the capsid, and then it has the nucleic acid on the inside, which is the RNA. Now, if I jump to the common cold, there are two specific types of the common cold, or common types, called the adenovirus and rhinovirus. You do need to know those specific types. You can see that the adenovirus and rhinovirus basically contains DNA. It's polyhedral. It does have attachment receptors that will attach to our cell, and then it will go into our cell. Here we have the flu. The flu is called the orthomyxovirus. The flu is actually short for influenza, and it actually is a kidney bean shape. It does have little glycoproteins on the outside, little receptors, and it does have RNA inside. Now, it does have a capsid around the RNA instead of outside the whole virus, but this is what the flu looks like. This is in a electron microscope, as it's identified in nanometers, very tiny. So the electron microscope many times shows up in black and white and gray scales as well. You can see the little stuffed animal there. If you go to giantmicrobes.com, you can actually buy a little stuffed animal of the flu or the cold, many different things. You can buy a little stuffed animal for athlete's foot. Okay, here's the T4 bacteriophage. The T4 bacteriophage is very small. You can see right here is the electron microscope. The electron microscope is going to be seen in shades of black and white and gray scale. The T4 bacteriophage is a virus, not a bacteria. It's a virus that attacks and kills bacteria. So you can see that in the word bacterio. Okay, that's what it attacks, and phage means to eat. Now, phagocytosis is going to be a term you're going to learn next unit, and that means to eat the cell or engulf the cell. You can see in this virus, it has a capsid, it has DNA inside, it has tail fibers or attachment receptors in this structure, very detailed in shape. You can see the T4 bacteriophage attack to a bacteria, so you can see how much larger the bacteria is. And scientists have used the T4 bacteriophage to stop very harmful bacteria. And this can be used instead of antibiotics. Now, here's a term that you'll need to know next chapter. Antibiotics is a medicine that will kill bacteria. Okay, not viruses, but antibiotics will be used to kill bacteria. Now, a vaccine does not kill a virus. It just helps your immune system. Now, doctors may also give you medicine with the antibiotics if you're trying to fight a bacterial disease to help your immune system and also kill a bacteria. But make sure you know the difference. Antibiotics kill bacteria. Antibiotics will not kill viruses. Okay, you need your immune system to be built up. So let's talk about the reproduction of viruses. There's two cycles that you need to know. The first one is the lytic cycle, and the second one is the lysogenic cycle. Okay, now let's talk about the reproduction of viruses. We have the lytic cycle, lysogenic cycle, and endocytosis. The lytic cycle is generally talking about the T4 bacteriophage, as well as the lysogenic cycle. The lytic cycle is when the T4 bacteriophage attaches to the host, it will inject its DNA into the host and then basically causing the host to replicate the DNA. The host cell will then explode and make more viruses. So the key here is the virus will be attached on the outside, inject the DNA, and then it will be non-functional and then float off. Now, the lysogenic cycle is just like the lytic cycle, except it doesn't, the DNA that the virus injects in doesn't start to kill the host. It just lays dormant. Okay, it doesn't kill the host yet. It will switch on maybe 2 years from now, and then it will start killing the host. Now, the last one is endocytosis. This is very common in animal viruses or viruses that attack humans. This is when the virus, such as the flu or the common cold that has those little attachment receptors, it will attach to our cell and then go inside. So remember, endo meaning going in, cyte meaning cell. So basically the virus will attach and then the whole virus will go inside. So let's take a look at some pictures of those to help you understand this a little more. Okay, the lytic cycle has 5 steps. As we said, the virus will attach to the host cell. It will then, step 2, penetrate the DNA into the host cell. It will inject the DNA into the host cell. Now, if you're using synonyms to identify these steps, that's totally fine. So instead of penetration, if you say injection, that's fine. It will inject the DNA or RNA into the host cell. Then the host cell is turned into that zombie mode. It no longer does its own life function. It replicates and makes different parts of the virus through its own transcription and translation processes. So it will make DNA or RNA or different pieces of the virus. It will then put those pieces together in step 4, assembly. And then step 5, it will release. The host cell will burst open, and all these new viruses that are made will then attack more cells. Here's attachment. Here we have the injection or penetration of the DNA. The virus now is non-functional. It can just float off. The host cell, and this is a bacterial cell, starts making more and more DNA, starts making the pieces. Then we move into assembly, step 4. And then we have step 5, release. The whole cell dies. And then now we have 4, then this will attach to 4 new bacteria. Okay, here's another picture of the lytic cycle. Here we have the attachment, injection, replication. Here they use the word synthesis instead of replication, assembly, and then release. Now the lysogenic cycle, as I said, is very similar to the lytic cycle, except it doesn't go right into killing the host. It doesn't go right into the host replicating the DNA yet. You'll still have attachment, you'll still have penetration, but that DNA will then weave itself into the host DNA and lay dormant. So at some point, it will then wake up and then cause replication. So we have step 3. This is the key. Make sure you highlight or understand that. Integration. The merging of the DNA that will lay dormant. So here we can see a picture, the attachment, the penetration, and then here we have the little red piece. You can see the little red is the viral DNA that merges into the host DNA, and it waits for the cell maybe to go through binary fission, and it's just many cells now have this host cell. So instead of attacking one and then having its offspring to attack many others, it's waiting for the bacteria to make hundreds through binary fission. Then it will wake up and then attack all of them because the information's in all of them. So you can see the contrast and the difference between the lytic cycle and lysogenic cycle. And now we're going to move into a virus attacking animal cells. So the key word is endocytosis. The virus attaches to the host cell. It then goes into the host cell, and when it's in the host cell, it will release its DNA. Do you need to know all the little names of these steps here, such as acidification? No. Just know what's going on in this process.